Hello, welcome to Science Last Week from Science Mission. I'm Sadashiva Pai and going to discuss three papers published in the week of April 28th. Let's start. First, we are going to discuss a paper published by Chris Knight's lab in Nature Communications. The paper talks about how mutation rate in bacteria is controlled. The findings in this paper are very important for health and diseases as authors identify a new mechanism by which bacterial mutation rate could be controlled. Findings may be important in controlling bacterial infection in disease states. Let's look at some of the literature. Mutation rate is a fundamental factor in evolutionary genetics. In nature, mutation rates are typically minimized as far as population genetic constraints allow. Rates of spontaneous mutation can vary both between and locally within genotypes. In E. coli, abundant variation in mutation rates is observed among natural isolates. Whether and how mutation rates at any particular site might vary, that is mutation rate plasticity MRP for a given genotype, is of particular interest. MRP can increase during environmental or genetic stress. Using E. coli, authors identify plasticity in the rate of mutation to rifampicin resistance. Authors determined mutation rate plasticity in E. coli K12 using fluctuation assays. These assays identify mutational events in RPOB gene by counting cells resistant to the antibiotic rifampicin arising in the absence of rifampicin. As shown in figure A, providing different amounts of nutrient allow cells to achieve different numbers of generations per day, that is, different absolute fitness. Authors found that variation of mutation rate related to changing absolute fitness. Mutation rate doubles with every reduction in absolute fitness by 2.6. Absolute fitness is affected by many independent variables or parameters, including culture volume, inoculum size, viability, productivity, and nutrient availability. Using various conditions by manipulating the strains paired, ancestral or evolved, culture volume, and culture period, authors eliminated various hypotheses and fitted a linear model for mutation rate with population density D, as shown in bottom figure. In other words, they found strong evidence for an inverse relationship between final population density on mutation rate. Less cells, more mutations. Authors wanted to identify molecular mechanism of this inverse relationship population density and mutation rate. luxs is a quorum sensing gene in bacteria and is responsible for various density related behaviors. So authors wanted to identify the role of luxs in MRP. They used E. coli K12 null luxs mutant, that is removing lux gene, both alone and in co-culture with ancestral B era strain. The null luxx mutant grows to approximately the same density as the K12 wild type and has an average a mutation rate that is not significantly different. However, they observed a significant difference in the slope of mutation rate in response to density between strains as shown in figure A. Furthermore, the null luxx mutant showed a response that is slope not significantly different from 0, indicating that identified MRP is not only density dependent but also luxs dependent. Authors also show that supplying aspartate to the medium at a level known to complement metabolic defects of the null luxs mutant restores the density dependence to levels seen in the wild type, the bottom figure. So authors conclude that metabolic product of luxs and not signaling molecule affects MRP. To test explicitly whether the effect of the null-less mutation acts via cell-cell interactions, authors co-cultured K12 cells marked via chloramphenicol resistance either with wild-type K12 or null luxs mutants. Authors then measured mutation rates of rifampicin resistance in marked fraction of the population. They found that populations comprising either combination of strains grow to very similar final population densities. However, mutation rate to rifampicin R depends strongly on the identity of the co-cultured strain, not on overall population density D. Average mutation rate increases by over a third in the presence of null luxs mutant relative to the presence of wild-type cells. Authors also found that effect of the null luxs mutant may be functionally complemented by aspartate in the medium, with an increase in average mutation rate. This confirms the action of luxs via cell-cell interaction and thus the activation via methyl cycle. Taken together, it is clear that MRP is dependent upon the social environment provided by luxs-dependent processes. Next, let's look at an interesting paper from Branwen's lab. This paper talks about how maternal food intake prior to the conception results in epigenetic modification in their progeny. Let's look at what is known about epigenetic control of progeny. Evidence is accumulating that environmental factors during early life have long-term effects on later health outcomes, and that these processes reflect epigenetic responses to periconceptional exposures. Metastable epialleles, that is MEs, are genomic regions at which DNA methylation is established in the early embryo and then stably maintained in differentiated tissues, leading to inter-individual epigenetic variation that affects multiple cell types. In experimental animals, maternal diet during the periconceptional period influences the establishment of DNA methylation at metastable epialleles in the offspring, with permanent phenotypic consequences. Several differences in the diet of rural Gambian women allowed the authors to test the hypothesis in humans. Methylation of MEs in peripheral blood lymphocytes, that is PBL, could be used as a readout of epigenetic state in other tissues. Authors measured plasma concentrations of key methyl donor pathway substrates. 8 of the 13 biomarkers and 2 derived variables showed significant differences between seasons of conception. Maternal periconceptional concentrations of folic acid, vitamin B2, methionine, betaine not shown here, and the ratio of S-adenosine methionine and S-adenosine homocysteine, and the ratio of betaine and dimethylglycine were higher in rainy season, and concentrations of ActB2, dimethylglycine, homocysteine, and S-adenosine homocysteine were lower. Next, authors tested whether these nutritionally driven seasonal differences in maternal periconceptional 1-carbon metabolism affect the establishment of DNA methylation at human MEs. The methylation analysis focused on several total loci. PBL and hair follicle samples, that is HF, of mesodermal and ectodermal tissues respectively from 2 to 8-month-old infants born to mothers in the main group were used. Percent methylation in HF and PBL within each of the remaining 6 MEs was highly correlated. Importantly, effects of season of conception on DNA methylation were also correlated in 2 tissue groups. Offspring of rainy season conceptions had higher levels of CpG methylation at the 6 MEs in PBL, and overall effect of season was highly significant. The seasonal pattern in HF samples was similar. So this is the first in human confirmation that the maternal blood biomarker status of substrates and cofactors required for methyl donor pathways measured around the time of conception predicts the methylation patterns of MEs in offspring. So what mother eats before conception is important and plays a role in the offspring's genetic information. Next, let's have a look at the paper published from Lucas's lab. The paper talks about using platelet serotonin transporter as a measure of understanding brain activity. Let's have a look at the literature. Serotonin transporter is abundantly expressed in humans, and gene SLC6A4 codes for serotonin transporter. Serotonin transporter removes serotonin from extracellular space. It has been postulated that there is a relationship between platelet and synaptosomal serotonin reuptake. But it is not known if the platelet serotonin uptake can predict neural activation of human brain networks. The results in this paper provides a tool to evaluate brain activity, to understand and also to do the efficacy of serotonin transporter inhibitors in depression. To evaluate potential impact of platelet Vmax on coherent functional networks, authors calculated functional connectivity, which provides a robust measure to assess temporal correlations in the functional MRI signal across functionally linked areas. Functional connectivity analysis explores which parts of the brain are coupled with a given seed region by cross-correlating the time series of the seed with all the other voxels. Figures display right hemispheric surface mapping of a whole brain correlation analysis between platelet Vmax and blood oxygen level dependent, that is bold activity. Significant brain areas showed positive and negative correlation. Negatively correlated clusters comprised areas of the default mode network, that is DMN, such as regions within the medial prefrontal cortex, anterior cingulate cortex, as well as posterior cingulate cortex, middle temporal gyrus, and inferior temporal gyrus. Positive correlations were found in the frontoparietal cortex control system, that is the central executive network and salience network, with a significant cluster located in the right motor cortex and premotor cortex. Scatter plot on the left shows a negative relationship between platelet Vmax and bold activity averaged across medial prefrontal cortex cluster. Scatter plot on the right shows positive relationship between platelet Vmax and bold activity averaged across the motor cortex cluster. So this study demonstrates that platelet Vmax may be used as an useful tool to understand the brain activity under normal and altered conditions like depression. Thank you for watching Science Last Week from Science Mission.